Hello, everyone, and welcome to this American Society for Cellular and Computational Toxicology webinar. My name is Christy Sullivan. I'm the ASCCT Secretary. And first of all, I wanted to mention a little bit of housekeeping. The webinar is being recorded, and ASCCT members will have access to the recording shortly after the event. Also, your lines are muted to cut down on background noise. And after the presentation, there will be an opportunity to ask questions using the question function in the control panel. So if you have a question during or after the presentation, feel free to type your question into the question panel, and our speaker will answer it. You can also reach me using the chat box during the webinar if you have a technical issue. I also want to highlight our annual meeting. We have our 5th annual meeting coming up this year, September 29th and 30th in North Carolina. We're very, very excited for this meeting. Abstracts are going to be accepted until July 22nd, so please submit your abstracts. Most abstracts will be presented as posters. However, the organizing committee will select some abstracts to be presented as oral presentations. We have 2 days and 2 plenary speakers. The theme for day 1 is Read Across with speaker Thomas Hartung. And we'll have the bulk of the meeting that day, including a mentoring activity, a poster session, and the business meeting, as well as a reception that evening. And then on the second day, we'll have about a half to 3/4 day with a theme of pluripotent stem cells and predictive toxicology. And the speaker is Mahendra Rao. And we will have a closing panel on the regulatory applicability of stem cells. So visit our website, ascctalks.org, to find out more information and to find directions to register and submit your abstract. Today's webinar is Human Primary Cell-Based In Vitro Systems for Translational Drug Safety and Mechanisms of Toxicity, and it's presented by Dr. Ellen Berg. Ellen Berg is Chief Scientific Officer at BioSeq, which is the division of DiscoverX, and is responsible for the company's BioMAP platform of human primary cell-based assays and predictive analysis tools. Her background includes the discovery and development of anti-inflammatory therapeutics, discovery program management, translational biology, and primary cell-based assay development and screening. Her research interests include phenotypic drug discovery, systems biology, drug and toxicity mechanism of action, and predictive methods for human efficacy and safety outcomes. And with that, I will turn it over to Dr. Berg.
So thank you, Christy. Thank you to the ASCCT for the invitation today to speak to you about advances in vitro testing for human toxicity and preclinical safety. And I hope to convince you today that phenotypic assays provide a valuable approach. So let's see. There we go. Our goal, of course, is improving drug safety, and despite our use of animal models and large panels of safety pharmacology assays, still some 20 to 30% of drugs that enter clinical trials fail due to safety. And what we believe this is needed, because the mechanisms of toxicity are so diverse and many remain unknown, that there is a need for a more modern, evidence-based approach to preclinical safety. So it's time. We know target-based assays are limited. We already have target-based assays in place, the ones we have. In vitro cell culture methods have advanced. There are now availability of cells and culture methods, automation, high throughput. And phenotypic assays do enable the discovery then of new mechanisms, potentially new targets, and a new better understanding of mechanisms of toxicity. So, but there's still challenges for developing in vitro phenotypic assays. We know human biology is extraordinarily complex. There are multiple levels of organization, from the molecular to pathways to different cell types we have to consider, and cell types interacting together into tissues and organs, etc. The systems are very dynamic. They're full of feedback mechanisms. So it makes assay design pretty hard. What cell types do we select? What endpoints do we measure? And how are we going to validate these assays? So our solution has been to focus on gold standards for the selection of the cell types that we're going to use in vitro assays, for the endpoints that we've chosen to measure, and the approach we've taken to build on our method of validating assays, building a large reference database that will provide comprehensive validation. So why human primary cells as the cell type of choice? These cells, they're early passage, they haven't been exposed to tissue culture plastic for very long. They are the most physiologically relevant in vitro cell we can get. They retain more of the feedback mechanisms than cell lines, and keep in mind that they certainly would have less adaptation to cell culture than stem cell derived cells, which spend weeks and weeks being differentiated in culture. And of course, assay systems that have co-cultures that allow you to measure interaction between cell types would be even more relevant. And human cell-based systems, of course, will be able to be compared directly to humans because there's no species differences to consider. So why have we selected clinical biomarkers as the endpoints to measure? These are the most proximal endpoints that we can get closest to clinical outcomes. Protein biomarkers, expression is often confirmed by histopathology. We know are validated in human clinical studies. They're known to be disease relevant. We understand how they respond to drugs. And particular cell surfaces secreted forms of these proteins are the substances that mediate tissue communication, the communication between cells and between tissues, and therefore provide a natural bridge between in vitro and in vivo. Finally, comprehensive validation is necessary to build enough confidence in our assays to make decisions. Through replicates and testing of large numbers of well-characterized reference compounds, once we have a large enough database, this facilitates the discovery of patterns through data mining, which then will allow predictive tests to be developed, as we'll discuss later in the talk. So for these, how do we put this all together? In our system, the profiling platform, the BioMAP platform that I'm going to talk to you today, starts with cells. We combine different cell types stimulated with activators, and these are maybe proteins that the cell types don't measure, but they would be exposed to in vivo, so we can add them exogenously. And then we validate these for the expression of the clinical biomarkers of interest. Of course, not just a single biomarker, but a panel of biomarkers. It gives us a better picture. This process is iterative. We test different cell types. It's optimized, optimizing the combination of activating factors until the pattern of biomarkers matches what we see in human tissues. And we further validate with standard of care drugs that we know how they act in people until we have a system that models as best as we can the disease or tissue of interest. It covers enough of the cell types, the pathways, and endpoints that provide us with enough coverage of that particular aspect of biology. And of course, to really cover a broad range of human biology, multiple systems, each of which will model a particular cellular or tissue setting. So in the panel of assays here, this is our Diversity Plus panel. Each of these assay systems here models the adaptive responses to one or more human environmental conditions, such as stress, immune responses, bacterial, and viral stimulation. So what happens when we profile a compound or reference agent across this panel of assays? This is the activity profile we get with by testing the approved drug Trametinib, which is a MEK inhibitor approved for the treatment of metastatic melanoma. So it's an oncology drug, and oncology drugs, as we know, often have side effects, and the side effects for Trametinib are well known, include rash, diarrhea, cardiotoxicity. So to point out, so what does this profile look like? The BioMAP systems are shown along the top. The biomarker endpoints, these are the protein levels that we're measuring, are listed on the x-axis. In every assay system, we measure overt cytotoxicity because if we change, if cells that are dying, they may change the levels of protein biomarkers for a different reason than if they're not dying. We measure the levels of each protein in the presence or absence of the drug and take a log expression ratio. So if you see a peak above the line, it indicates that that particular biomarker endpoint level has increased in the presence of the drug here. And of course, if it's below the line, it's decreased. The four different colors here represent the activity profiles at different concentrations. And so you can see concentration-dependent effects as shown here. And even though we do see with many markers a concentration-dependent effect, overall, the shape of this profile over multiple concentrations as shown here is remarkably consistent. And this profile shape is a pattern that I'll get back to you later and tells us something about mechanism of action. So just to remind you that every single one of these endpoints that we're measuring in these systems is a clinically relevant biomarker and means something. So what do the biomarkers that have changed in the presence of Trametinib here mean? Well, first of all, there are biomarker endpoints here that are consistent with the known role of MEK kinase in cell cycle, inhibition of endothelial cell proliferation, T cell, smooth muscle cell, and fibroblast proliferation. We also see changes in biomarkers that are involved in angiogenesis. We see changes in a number of biomarkers that are involved in tissue remodeling. These are matrix metalloproteases and plasminogen activator family members. All of these activities are relevant to the role of MEK kinase, the target, and the effect of the drug Trametinib in oncology patients. So these are all efficacy-related biomarkers, consistent with both. But we also see activities that are relevant to some of the side effects. Patients who are taking Trametinib are at increased risk of infection. We see inhibition of many biomarkers that are acute pro-inflammatory proteins like IL-1, TNF-alpha, and IL-8. We see upregulation in serum amyloid A. This is serum amyloid A is actually a risk factor for cardiovascular events. And we see upregulation of VCAM, vascular cell adhesion molecule. This is a pro-inflammatory adhesion molecule that recruits chronic inflammatory cells in sites of inflammation. And so all of these markers then are relevant to safety-related effects of Trametinib in the patient. So all of these markers and changes that we see with this drug in this in vitro system are correlated with known clinical effects of the drug. So you can imagine not only having data on this drug, but many other drugs and many other types of materials that we have tested across this platform gives us, extraordinary amount of power that underlies the platform. for mechanism prediction using data mining, for further correlations with both clinical and preclinical effects, and the ability to connect projects across therapeutic areas, because this is a standardized panel that represents basic human tissue biology. So what have we learned from analyzing patterns in the data from looking at this reference database? I'm going to talk about 3 types of patterns that we have been studying. The first is simple concentration response patterns. And this has yielded some insights into in vivo therapeutic windows. The other type of pattern are the entire profile pattern, which we just saw for the MEK inhibitor, Trametinib, and these can tell us something about mechanisms of action, target mechanisms of action. And then furthermore, the third type of pattern are subsignatures or profile motifs, maybe of just a couple of different activities that we have correlated with particular adverse drug reactions and are defined by particular mechanisms as well. I'm going to give you a few case studies on this last point. So in a way, these data sets can be applied for a biology-based read across, very similar to the way we use chemical structure features to do a chemical read across for trying to predict what you might expect with a new chemical. This is a way to further characterize agents and materials and be able to compare them head-to-head against a very large set of other reference agents that we know something about. So let me first talk about concentration response patterns. So here's an example of a particular type of response pattern that we see. This compound is very active and it has a sharp dose response. And what I mean by sharp, it means that it has a different pattern at every concentration that we test. And in fact, in this case, at the top concentration in red and actually probably in orange as well, this compound is overtly cytotoxic to multiple cell types and systems across the panel. So this is one type of a profile that you see. It goes very quickly from cytotoxicity and then down to almost inactivity. The other type, sometimes we see inactive agents obviously over a concentration range that we're testing. And we see very selective activity profiles as shown on the bottom left. And then we see the profiles like the one I'm showing here and the one we saw for Trametinib, in which the activity profile is very consistent and very stable over many concentrations. And this is a feature that we describe as being dose resistant, because the activity profile, it doesn't change over many wide range of concentrations. And we have been able to use these concentration response patterns to inform expectations for therapeutic windows in vivo. And here is a list of some approved drugs shown on the left and diverse mechanisms of action. And what we see on the right, these are the concentrations at which these are active and have a dose-resistant profile across the systems that I've been describing. And what we can see for fluticasone, it is active and has the same shape profile over multiple orders of magnitude of concentrations. And so it has a very wide-ranging therapeutic window here at these active non-cytotoxic concentrations. And something like cisplatin has a very much shorter activity window here, in vitro activity window here. And of course, what do we know about cisplatin? It has a very short therapeutic window in vivo as well. And when we overlay this with overt cytotoxicity, you can begin to calculate, a range of activity versus cytotoxicity, sort of a safe range. And then shown here in red, these are the clinical exposure levels. These are the average maximal plasma levels of these drugs that have been reported. Remember, an average is, the range is usually 50 to 100-fold. So there's quite a range among patients, and these are the average maximal concentrations shown here in red. And now you can see how this gives you an idea of the therapeutic window at which you'll have a safe, active compound without toxicity, and be able to, furthermore, we can use this to prioritize compounds for their the range of their therapeutic windows. And we know, for example, for the kinase inhibitors, that sorafenib and sunitinib, these are the compounds with very narrow therapeutic windows, more narrow than trametinib. In particular, sorafenib has very significant side effects, and trametinib, for example, has much fewer. So we can use this feature of this concentration response pattern to give us an indication of what to expect when we go in vivo in terms of what's going to happen in a tissue, assuming we reach a particular particular concentration in that tissue. So the other type of pattern that I want to talk about is a pattern that's across the entire profile. And we can use mathematical ways to look for similar or different patterns, correlation metrics such as Pearson, which depend more on shape of the profile, are particularly useful. And when we look at many compounds together, as shown on this next graph, we can see that based on analysis of profile shapes, we have compounds clustering as to whether, if they're very similar to each other, they would be clustered together here, and if they're very different from each other, they would cluster away from each other, as shown in the aqua. And the lines that are drawn between compounds indicate that the correlation, the pairwise correlation coefficient between those two profiles, is above a certain level. And you can see further here, this now is showing you some example profiles from these various mechanism classes, how really distinct these profiles are from each other. So they can get very discrete and very different from each other. And we have actually leveraged this feature to develop, to be able to use machine learning and develop automated classification algorithms for particular mechanisms. And this really starts with having multiple compounds that will be specific for the same target mechanism. For example, here are 3 p38 MAP kinase inhibitors, and you can see how their activity profile shapes are very similar to one another. In fact, you can identify really a consensus profile that reflects the target-specific biology. This is a p38 MAP kinase profile. But when we take the profiles from many compounds that we've tested at multiple concentrations for many different target mechanisms, we've been able to build classifiers for different mechanism classes, based on these activity profiles, and that perform, fairly well. And we published this a few years ago, but just to say that these mechanism classifiers perform pretty well, and we've actually developed 28 of these for particular mechanism classes. Of course, we need to have the compounds to build these classifiers. And just to note that many of these are relevant to safety targets that you might be aware of. And also to point out that in this case, some of these targets that are relevant for safety concerns in patients may also actually be important for efficacy. So calcineurin, this is important for immunomodulation, but immunomodulation or immunosuppression, but actually if you have an autoimmune disease, you want to suppress the immune system. So, and the same way microtubule disruptors are, this is a class of oncology drugs, and mitochondrial inhibitors, there are some diabetes drugs that are mitochondrial inhibitors. And so one has to be aware that every mechanism of toxicity may also be a mechanism for efficacy depending on the indication. So the third type of pattern analysis that I'm going to talk about next is really the discovery of profile motifs or signatures, subsignatures within the overall profile that we've identified as correlating with particular adverse drug reactions. I'm going to walk you through a couple of case studies, one related to thrombosis-related side effects, and the other having to do with mechanisms of skin irritation. I'll summarize a few other signatures and then close with some points on our future directions. So thrombosis-related side effects. This starts with the protein, the surface protein tissue factor, which is expressed on platelets and also expressed by endothelial cells. It's known to be an important player for normal human physiology and wound healing and angiogenesis. Tissue factor, CD142, is the primary cellular initiator of blood coagulation and thrombosis. It is involved in the recruitment of platelets into the tissue sites and promotes fibrin matrix deposition, which is important for repair. But it also has pathologic consequences, which can lead to aberrant coagulation and blood clots, which will give you clinical manifestations such as deep vein thrombosis, stroke, myocardial infarction, and pulmonary embolism. And in fact, Ebola actually causes a consumptive coagulopathy, which then leads, once you use up all of the coagulation enzymes, all the clotting enzymes, then you've got no more left, and you then hemorrhage. So it's a very important protein and molecule in vascular hemostasis. So what we noticed several years ago was that in one of our vascular inflammation models, we saw upregulation of cell surface tissue factor induced by inhibitors of mTOR. These are the limus drugs, and also aryl hydrocarbon receptor agonists. And this was a class of chemicals that we had tested as part of the EPA's ToxCast program. And we noted at the time that both of those are associated with thrombosis-related side effects. And so it led us to, in this particular model system, this is a vascular endothelial cells that are stimulated with pro-inflammatory cytokines. It's a model of vascular health response to infection and inflammation. And so this observation led us to go look at a few other drugs that we knew that were associated with thrombosis-related side effects, including the selective estrogen receptor modulators and oral contraceptives, a few antipsychotic drugs, and some oncology drugs. And what we had observed was that all of these drugs caused an increased level of tissue factor in our particular model system. Now, this is where the database, having a large reference database comes into play, allowing you to confirm an association of any signature activities with a particular side effect. And furthermore, this allowed us to uncover some of the mechanisms. So we start with a signature, we compare it against the database, and in this case, this particular signature, this increased level of tissue factor, in this particular system was relatively uncommon, and actually only a small percentage of compounds in our database at the time were found to have the signature. More interestingly was the fact that we were able, based on those few compounds, because there were a few compounds, they were enriched in particular mechanisms, and the mechanism classes are listed here. This actually lists all of the compounds that had the signature on the left, and the mechanisms here are shown on the right. And again, when looking through these mechanisms, what became apparent, what common feature of these particular mechanisms is that they were all involved in the process of autophagy. So autophagy is a normal cellular process by which the cell consumes itself and to form, to either generate, new nutrients, or to recycle materials. And it does this by initiating the nucleation of new vesicles, and then these vesicles ultimately consume either cytoplasm or various parts of the cell, and then the materials inside those vesicles get broken down and degraded by lysosomes, and then the products then become recycled into other parts of the cell. This is a process that's controlled by bacteria, by oxygen levels, by nutrient levels, by lipids, and it turns out that all of these agents shown in dark blue here are agents that affect the process of autophagy in a specific way. They increase the number of vesicles, and they do this either by increasing vesicle nucleation, so you get more vesicles produced, or by blocking the degradation of vesicles. And all of those lead to increased levels of tissue factor on the surface. And this actually has teleological implications, because what would happen if you increase the levels of tissue factor on the vascular surface? Well, you would recruit nutrient-rich platelets. So in times of nutrient deprivation, this is a natural response of the tissue to bring in more nutrition to the cell. So, but of course, when it goes wrong, that's where you end up with, overt activity of tissue factor and aberrant coagulation. So all of this information then can help us put together a framework for an adverse outcome pathway. So these pathways are very important for safety assessment, and now being used to be able to quantify risks. And so we can start with a molecular initiating event, something you could measure, activation of a target such as the aryl hydrocarbon receptor or inhibition of mTOR. And then one of the key events would be increased in autophagic vacuolization, and then subsequently upregulation of cell surface tissue factor, initiation of coagulation, and then at some incidence level, the occurrence of the clinical effect, which could be something like deep vein thrombosis. And so we now have an assay that now can be used to measure the effects of downstream of the molecular initiating events, which could be very, very, very helpful. So this project, we've got a predictive assay for thrombosis-related side effects. We found that this particular assay measures the effects on at least 12 different cellular targets and actually connects them through this very detailed mechanistic process of increasing autophagic vacuolization endothelial cells. The impact then is we're now able to use this information to develop a framework that we can now start to put to quantify and at some point be able to potential risk. For drugs in the clinical stage, this information might change how a clinical program is managed or how patients are stratified or managed. And of course, if you're earlier in a drug discovery process, you have the opportunity, if it's because of an off-target effect, to prioritize compounds that would have reduced side effects. So the second case study I'm going to walk you through is a predictive signature, sentinel signature for skin irritation and a potentially new mechanism of toxicity. So this starts with a project with a collaborator. They had some compounds in a CNS program, and the target, it was a known target-based program, GPCR target, and they had discontinued the program because of skin toxicity in non-human primates. And you can imagine the cost of non-human primate studies. Unknown mechanism of toxicity, and they had data from two compounds that showed effects in the primates. And their goal here then is, can we identify the mechanism of toxicity and determine whether it's on or off target? So when we profiled this compound, it was unexpectedly active across our panel, and we weren't particularly expecting this because we did not believe the target was expressed in these cell types. And when we compared the two compounds together, we identified, in particular, one model system where there were certainly shared activities between these two compounds: increased prostaglandin E2 and decreased TNF-alpha. And this particular system here is a co-culture with peripheral blood mononuclear cells and vascular endothelial cells. And this model system is stimulated through toll-like receptor ligands, giving you a biology model that includes monocyte activation, vascular inflammation, response to infection. So what was particularly interesting about this activity profile, the signature, is that we discovered that it was common among known irritants. So it was shared by chemical vesicants. These are blister agents, retinoids. These are agonists of retinoic acid receptors, and some VDR, vitamin D receptor agonists, calcitriol. All of these are known skin irritants. And it turns out that dermal prostaglandin E2 is actually a biomarker for skin irritation, and the damage from chemical vesicants has shown to be dependent on cyclooxygenase. This is the enzyme that is rate limiting for the production of prostaglandins. And so this points to PGE2, increased levels of PGE2 being particularly associated with these compounds and the mechanism of this activity of this compound. So we can further use other results from profiling the downstream mediators across our panels to put together a picture of how irritants act on the tissue to induce irritation responses. So if we start with the irritant, increasing prostaglandin E2 levels, changing collagen ratios, PGE2 then promotes vascular permeability as well as production of IL-6 and IL-17. IL-17 in turn increases IL-8 and tissue plasminogen activator. Tissue plasminogen activator then together with altered collagen-1 ratios is leading to a compromised barrier function. Increased IL-17, IL-8, and IL-6 all promote neutrophilic inflammation. And some irritants, including compound 47 and retinoids, also increase eotaxin 3. This is an adhesion molecule that promotes the recruitment of Th2 type inflammatory cells, such as eosinophils and mast cells. These are the cell types that are involved in allergic types of inflammation. And so we can see from analysis across these panels we can put together a better understanding of how this irritation response is occurring. So again, we can go back to our reference database to confirm the association of this signature with irritation, skin irritation, and use this large database to further uncover mechanisms of action. So we do this as we did before. Again, relatively uncommon signature, but interestingly, of the compounds that we identified, a few of these were drugs that had clinical information on them, and 14 of 15 of the drugs that did have information were reported to have skin side effects. So a pretty high association here. Furthermore, we could use these agents, and these are listed here in the next slide, all of the active agents, to infer the mechanisms that are associated with this signature. In addition to retinoic acid receptors and aryl hydrocarbon receptors, we also saw, and vitamin D receptors, we also saw certain modulation of aryl hydrocarbon receptor, PKC activation, and thromboxane A2 synthetase. And we've presented this data here earlier this year at SOT. So again, starting with a particular activity profile and association with a clinical side effect, having the database allows us to uncover associated target mechanisms of action. So what about compound 47? So it turns out when it was tested across nuclear hormone receptor transcriptional pathway assays, our collaborator identified induction that these compounds induce are active on the retinoic acid receptor, as well as potentially on aryl hydrocarbon receptor. So at the end of the day, again, we have a novel assay, in vitro phenotypic assay and signature that's associated with skin irritation. 5 target mechanisms were found to be involved, and the data allowed us to build a pretty in-depth model for how these irritants are causing the irritation response in the tissue. Impact particular collaborator is opportunity to rescue the program. Now we have an assay and can be used to identify backup compounds to potentially, next generation products with reduced side effects. And anytime we understand the mechanism that's behind a toxicity, it opens up how we might address mitigation strategies for drugs that are already being used in people and are approved. So these 2 signatures aren't the only signatures we've identified. We've identified several other signatures that are associated with particular adverse effects, some in preclinical species, and most of these in humans. Not only do we have reference compounds that are induced, that have these signatures or are associated with this, but we've used our reference database to pinpoint and identify, potential targets and pathways that are implicated for each one of these. And so these are not black boxes assays. These really are mechanism-based signatures, which allow you, if you see this in a program or a molecule that you're testing, immediately be able to go to, a small set of confirmatory studies or other target-based assays to be used for further development in further use in programs. So, in summary, human in vitro phenotypic assays can offer a lot in terms of utility for safety and toxicology applications. They can be built to cover a broad range of human biology and mechanisms. They can be standardized. They're sufficiently reproducible to be able to classify, to use data to classify mechanisms and develop predictors using machine learning. And our selection of clinical biomarkers as endpoints and the design of these assays, facilitates integration of this data with, discovery, preclinical, and clinical safety information. So, in conclusion, I hope you'll agree with me, it's time to implement in vitro phenotypic assays and preclinical safety. Animal tests aren't predictive enough. Our current safety panels are insufficient. We still need more mechanisms and a better understanding of toxicities. In vitro cell culture methods have advanced. There are, we have the availability of cells, automation, and sufficient experience now to have validated these methods. Phenotypic assays in particular are useful because they enable discovery of new mechanisms and a new understanding of the biology of toxicity outcomes. Phenotypic assays can capture activities and integrate information from many different targets, so they can connect multiple targets to the same clinical outcome. Not only does it help us with understanding the biology, but this information is very useful for construction of adverse outcome pathways that can be used ultimately in safety assessment. So in terms of future directions, there are certainly challenges remaining. For predicting human toxicities, one of the most challenging aspects is the fact that there's really no ground truth. Human toxicity data is very inconsistently reported. There are few standards. There's always missing information on patients as well as exposure levels. And if you don't have a ground truth, you cannot build a predictive model. Well, you can, but they're not very meaningful. If you don't have the data, you don't know what your true positive and true negative rates are. So I believe going forward that there will be a role for health IoT. By continuous biomonitoring of safety biomarkers, we will get the data. We will be able to standardize safety monitoring, correlate with exposure data, and build the data sets that are required for us to prove or disprove how our in vitro assays, how predictive are our in vitro assays for human clinical outcomes. And so with that, I'd like to acknowledge my team at BioSeq here in biology, our analytics group, on our operations team, as well as collaborators and at Pfizer, at the EPA, at the NIEHS, Safer Medicines Trust, Christy, of course, at the Physicians Committee for Responsible Medicine, and my collaborators in phenotypic drug discovery from Eli Lilly. And with that, I'll close and take any questions.
Okay, thank you, Dr. Berg. That was a wonderful overview of the work that you've been doing. And I especially like the last slide about, next steps and how we can integrate this type of information into the drug discovery process. I don't see any questions so far. If you do have a question, please type it into the chat box, the question box. But in case someone is typing, I'm going to ask, regarding your statement that we need to be able to ground truth these assays with clinical data, are you having early success with partnerships with clinicians or with clinical study directors? or even with the FDA trying to partner together to get some of that clinical biomarker data?
I think we're at early stages.
Yeah.
The biomarkers that are measured in clinical studies right now, they don't overlap with what we're measuring. What is missing is the details of toxicities. There's been certainly some standardization in terms of what we measure and how do we classify, serious side effects, serious adverse effects, and all of that. But what is missing is, at the patient level. So we'll get aggregated information and missing information. For example, in skin side effects and skin toxicity, it's pathologists have different ways they describe skin reactions, and so that's it's a problem. So the data that are from an individual clinical trial within itself is great, but between clinical trials is where we're having or where we have the problems. I think now that so many companies use CROs to run clinical trials, it's such a team effort to get a clinical trial done now. I think we all have to work together and set up the rules and how we're going to measure these things going forward. Anytime you add some other step to a clinical trial, just increases the cost, and that's a problem. So how do we get the information we need that's cost efficient? And that's really why I do think these biosensors that people are wearing these days someday will be the way forward, because then the individual patient or the person is motivated to comply, because and we can standardize what's being measured and how the data is being handled and captured. So that's really where I think things are going.
And possibly even more so with post-marketing surveillance, that consistency and sort of automatic reporting is even more important, I would think.
Yes, it's fairly inconsistent now.
Yeah.
It's the same way you comment about a shopping experience. We're not consistent in our complaints or our about events. The other issue is we don't have exposure information on post-marketing. So we don't know how much drug the patient, we may know, the patient may report how much drug they were taking, their dose, but we don't know what they were exposed. So lack of good, pharmacokinetic data exposure data is a challenge as well, because we know exposure drives toxicity.
Right. So we do have a question from Michael Batchelor. He's wondering, how widespread is phenotypic screening currently in use within the major pharmaceutical companies?
That's a great question. I know in phenotypic drug discovery, it's about 10 to 20% of the screens being done across pharmaceutical companies is phenotypic. And if you think about it in safety, anytime you do a toxicity assay, that's a phenotypic assay, because there are many mechanisms that can cause cell death. Right. So very widespread then. But I think what we're looking for is more valuable assays. Toxicity to humans is not about cell death, right? Because you can cause toxicity without, really with mechanisms that are, destroy the system, but not necessarily a particular cell type.
Right. I think it's an interesting question, and we in the chemical arena, we're also trying to figure this out, especially with adverse outcome pathways starting to be more commonly used, and trying to weigh the advantages and disadvantages of the single readout assays versus a phenotypic assay that may cover multiple mechanisms and multiple adverse outcome pathways. You're from an efficiency standpoint, that would be more useful. So it's just really defining your question that I think is the most important more important thing to do once you have access to different types of assays.
And I think we have to be evidence-based, and we have to have very high bar for validation of assays. I read so many papers where they pick and choose what the compounds they're going to use to validate their assay, and that's just not good enough. It has to be extraordinarily well characterized, because that's the only way we're going to get to the level of confidence that we need.
Right. So I don't see any other questions, so I think I'm going to close out the webinar, and feel free to email me afterwards if you think of a question or have any feedback. And otherwise, I will thank Dr. Berg for her time and presentation, and thank you all for your time as well.
All right, and thank you everyone for your attention as well.
Have a great day, everyone. Bye-bye.